Hello everyone. So my STEAM fair project is about pH and the growth of the garlic ball, and I will also examine it through the cell cycle under microscope. So I organize my slideshow through this content, where I start with the background research and end with citation. So pre-experiment. I did some background research before I start my experiment, so I found out that pH is the scale that used to specify how acidic or basic a water-based solution is. So for example, for human blood, the pH is between 7.35 to 7.45, which is slightly basic, and on the pH strip, the color of slightly basic will show the color of greenish color. If it is strong basic, it will show a purple color or blue color. And for lemon, the pH is 2, which is acidic, and on the pH test strip, it will show a reddish color or orange color. So pH is the fundamental element for living organism, and it is very important for the organism who grow either in water or soil. My hypothesis is if the pH of the living condition of the garlic balls change, the growth of root of the garlic ball will also be limited, which results in a self-cycle. So basically, I think that changing pH will limit the growth. And I list my variables. My independent variable is the pH of the living environment, and my dependent variable is the growth of the roots that measure in a milliliter and the imaging that under microscope. My control variable is the amount of sunlight, amount of water, and type of onion. The control group will be the garlic ball that grow under a neutral condition. The experimental group will be the garlic balls that grows under a acidic condition and alkaline condition or basic condition. My experimental unit is the garlic ball that grow in each water bottles. So there is 3 sample in total. Here's my material. For some of them is laboratory material, and for example like hydrochloric acid and carnauba fluid and acetone carmine solution, and they will help the cell to fix the DNA, and so we can preserve the shape and the overall structure of the cell. Before we go through the procedure, we need to deploy the solution first. So since we have 3 water bottles, which means we need to deploy 3 solutions. The first one is the acidic solution, which I use hydrochloric acid to lower down the pH of the water. And the pH is 2.5 at the end of my measurement. And the next solution is the neutral solution, which has the pH of 7. Since water is already neutral, so you just add water in the water bottle is fine. And the third solution is alkaline solution or the basic solution that has pH of 8.2, and I use the baking soda or the sodium bicarbonate to change the water to slightly basic. And the procedure is quite long, and I will simplify it to you grow the garlic ball and you will use the root of the garlic ball and you put it into the carnauba fluid to let the fluid fix the DNA and the cell structure, and then you use dye to make the cell structure visible under the microscope, and that's the overall procedure is about. Post experiment. So here is the graph for each garlic ball under 3 conditions, and as the result we can see that the neutral condition, that the garlic ball that grew under a neutral condition had the greatest growth compared to the alkaline and acidic condition. I will start with the garlible that grow under the acidic environment. Because there's no growth of the roots that the garlible grow under an acidic environment, which make the experiment unable to proceed. So it's one of the evidence that lowering the pH to 2.5 will limit the growth of the garlible. And I have the picture of my garlible at the last day of my experiment, which you can see that there's no any growth. And the garlible grew under a neutral environment, have a healthy root, and after 5 days, and these roots are qualified to proceed the experiment, and the imaging under the microscope shows a clear cell cycle. Here is the graph for the growth of the root, and as you can see, the picture shows that there is the growth of root of the garlible that grew under the neutral condition. And because we can use the root, so we can able to see the cellular structure under microscope. For a quick reminder that in the cell cycle, most of the time the cells stay in the interface, which is growth and DNA replication. And the main thing that I will focus today is about the mitosis and cytokinesis. So as you can see in the interface, the cells is only showing the cell wall and membrane and also nucleus. And the interface contains G1 phase DNA replication and G2 phase. So start at the early prophase is the beginning of the mitosis. I split prophase into two parts because I think they look quite differently from each other. So in the early prophase, we can see the chromosomes condense and become visible with the white circle in the middle of the cell. And the late prophase, the nuclear envelope breaks down, so now you cannot see the white part in the middle of the cell because the nuclear envelope breaks down. And the spindle fiber is attached to each chromosome, and the chromosome condense. And the following stage is the metaphase, and where the chromosomes they are lining up in the middle of the plate, and each sister chromatid is attached to the spindle fiber from opposite poles. So you can see that the sister chromatids they are lining up in the middle of the cell, and they are ready to pull apart in the anaphase. And compare metaphase to anaphase, you can see clear differences is that the spinal fiber and the microtubule already pulls the sister chromatid apart to chromosomes. So in anaphase, the centromere split and the microtubule from the opposite side pulls the chromosomes toward each pole. And then we have the telophase, which is when the chromosome reach to the opposite poles and begin to decondense. So in here, the nuclear membrane start to appear again, but the spindle fiber keep pulling the poles apart. So it will still keep pulling the chromosome to the opposite pole until the cytokinesis started. So cytokinesis is not part of the mitosis. It will start at the end of telophase, and this is the part when two cells separated from each other, which is when the cell wall separates the two daughter cells completely. And as a result, we know that mitosis produces identical cells or identical daughter cells that they look exactly the same to the original cell and to each other. And for the garlible that grow under an alkaline condition, which the result is not really successful, but there is still some growth of root after 5 days. So since there is a root, then I will continue the experiment with the limited root. The imaging under the microscope do not show a clear cell cycle. So here, according to the picture, you can see that there's only tiny growth of the root for the garlic bulb that grew under alkaline condition. And we can see that all the images, they are not really clear as the imaging from the neutral condition, because the alkaline condition will affect the cell structure of the root, and which made the root weaker to resist the erosion from the hydrochloric acid and from other chemical that we need to apply before we start to examine it under the microscope. So this root, the cell structure of this root are partially destroyed by the chemical, but we still can see the overall structure from the microscope, since the imaging one shows a cell with the overall shape but not clear activity inside the cell. So conclusion for my experiment will be that the result of the experiment support my hypothesis that change of pH will limit the growth of the root of the garlic bulb. So the acidic condition produce no growth, and because there's no growth, so the experiment cannot proceed, because the low acidic will damage the cell structure and ability to reproduce. Neutral condition produce a healthy root, and this root can be examined through the microscope and see the clear cell cycle. The garlic bulb that grow under alkaline condition produce few roots that are very weak and have partially destroyed cellular structure by the basic condition. And as the chemical also impact the cellular structure, so which result the imaging is not clear. And there's an improvement that I can make next time. So since I only picked 2 pH value from the pH scale, so for my conclusion, I can include more pH values, such as I can include pH of 3, 4, 5, 6, 7, 8, 9, 10, all of them, and make comparative data to each other, and which is more reliable and more valid, because my range for my pH is too far away. For the acidic condition, the 2.5 pH is too far away from the neutral, but for the basic condition, the 8.5 is fairly close to 7. Also, repeat trials can increase the reliability of the result. And also, you can use different plants to apply the same experiment. They can construct a comparison among different species. So some species might favor in slightly basic condition or slightly acidic condition, which you might find out by using different species. And I also relate this experiment to the real world, that farmer will add the chemical to the soil to change the pH of the soil and make the condition more suitable for seed to germinate. For example, some farmer will use this type of limestone to neutralize the pH of the soil while the soil is lack of magnesium, which is one of the fundamental nutrients of living organisms. To balance or neutralize the pH of the soil to the ideal condition can help farmer yield more product and crops. And here's my and here's my citation and the end. Thank you for your listening. And if you have any questions, and you're welcome to text me.